Assalamu alaikum. Today's lecture about pancreatic hormones. Your recommended chapter is from IdiTEN textbook is chapter 79. The pancreas composed of two parts: the exocrine part, which is the acini that secrete the bicarbonate and the digestive enzymes, and the endocrine parts, which is a collection of cells. We call them islets of Langerhans. Around 1 to 2 million of these islets found in the pancreas. In that islets, there are different cells that secrete different types of hormones. The first more abundant cell, which is around 60% of the islets, this is the gray colored one, these are the beta cells, and these secretes insulin and amylin. Amylin, the functional properties of it is not well known, but it is secreted with insulin almost the same amount. Then we have the second cells, the alpha cells. These are the blue one, which is around 25% of the islets, and they secrete the hormone glucagon. And then we have the delta cells, which around 10% of the islets, and they secrete the hormone somatostatin. And there are very few cells, we call them BP cells, or that secrete the hormone pancreatic polypeptide. One of the most important hormones secreted by pancreas is insulin. You can consider insulin as one of the most important medical discoveries in the 20th century, which is around 1921 it was discovered, and by 1922 they have been purified in a form that can be given to a patient. And since that, the life expectancy of patient with type 1 diabetes improved, because this is the treatment for type 1 diabetes, because their pancreas cannot produce insulin, so they take it as an injectable form. As you see from the picture here, we can consider the insulin as the key for the glucose to enter the cell. But also, as we will see later, that the insulin play a very important role in protein metabolism and fat metabolism. From this slide, you can see the biosynthesis of insulin. So insulin is a protein, so it is similar machinery of protein synthesis in the other cells. So at the beginning it will be formed as a big protein molecule, which we call preproinsulin. The preproinsulin composed of 4 subunits. The A and the B chains, they are the one which form the insulin later on, as we will see, and this is the N-terminal signal peptide, and this is a connecting peptide that connects A and the B chain. Inside the endoplasmic reticulum, this N-terminal signal peptide will be removed, and the A and the B chains will be bound together by disulfide bridges, and the connecting peptide still there, and this is what we call proinsulin. Then in the Golgi apparatus, this C peptide will be removed and the insulin will be formed, and this is the active insulin. So it's composed of alpha and beta polypeptide chain bound with a disulfide bonds, and the C peptide has no physiological effect. They are both released to the circulation and in an equimolar amount. The insulin has a very short half-life because it will be cleared from the circulation, and it's only the one which is bound cleared in circulation because it is not bound to plasma proteins. and the effective one is the one which will bind to the receptor, and this is very important for the function of insulin. The C-peptide, the most important use of the C-peptide, because I told you they are both released from the pancreas in an equimolar amount, so it is used as a marker of the natural insulin synthesis and secretion. So patient with diabetes, if you want to check how much insulin, natural insulin secreted by the pancreas, you can measure the C-peptide. For the insulin to initiate its effect on the target cell, it has to bind to its receptor. Insulin receptor is a very big protein molecule composed of 4 subunits. 2 alpha subunits, they are located outside the cell, and 2 beta subunits that are going through the cell membrane and protruding inside the cytoplasm. So once the insulin is bind to the alpha subunits outside the cell, because they are all linked together, this will cause autophosphorylation of the beta subunit, the protruding part of the beta subunits, and then this beta subunits is attached to an enzyme called tyrosine kinase. So the autophosphorylation of the beta subunit will activate the enzyme tyrosine kinase, and which then will phosphorylate a group of enzymes. They are called, some of them they are called insulin receptor substrates. So the phosphorylation of these enzymes, some of them will be active and some of them will be inactive, and this will cause an effect on different aspects of metabolism of the carbohydrate, the fat, the protein, and even there is a long-term effect on growth and gene expression. So once the insulin binds to the receptors, the main end effects of this insulin can be shown immediately, what we call an immediate action of insulin, and one of these immediate actions is the increase the uptake of the glucose by the cells. So the insulin will cause translocation of vesicles filled with transporters. They are called GLUT4 transporters. They will be translocated into the cell membrane, where they be embedded in the cell membrane, and this will allow the glucose to enter the cells through these GLUT4 receptors, which are located in most of the cells with very few exceptions. Another immediate action or a rapid action of insulin is the increased permeability of the cell to the amino acids and potassium and phosphate, which then will increase their transport of these molecules to inside the cell. So it will increase the cell membrane permeability to amino acid and potassium. And this is the reason why insulin sometimes used in treating patients with hyperkalemia, because one of the immediate actions of insulin is to increase the cell membrane permeability to potassium, which will clear the potassium from the blood by increasing its entrance inside the cells. There are some slower effects occur because of insulin, like within 10 to 15 minutes, and this is mainly related to change in the phosphorylation states of some enzymes, which their action might take few minutes to get effective. Another much slower effect, which might take hours or even days, this is the change in the rate of messenger RNA translation to proteins, and also the how much of messenger RNA produced from the DNA. So these things will take longer time. What is the mechanism of beta cells in secreting insulin? One of the main controllers of insulin secretion is the glucose level in the blood. So when we have a high glucose in the blood, this will be will enter the beta cells through a different type of glucose transporter, which is GLUT2. So then the type of glucose transporter in the beta cells is GLUT2, not GLUT4 like the other cells of the body. So once the glucose enter the cell through glucokinase, it will be converted to glucose 6-phosphate, which then will undergo oxidation, and this will increase the level of ATP. The increase in ATP will shut down or close the ATP potassium channels, which will prevent potassium from going inside, and this will cause depolarization of the cell membrane, which will affect the voltage gated calcium channels, which will cause them to open, and the calcium will go inside the cells. Once the calcium increase inside the cell, this will cause translocation of these vesicles filled with insulin, and then they will release the insulin into the blood. One of the most important effects of insulin is on carbohydrate metabolism. I'm sure everybody of us loves the smell of carbohydrate, but it has a major consequences. So once we have a high carbohydrate meal, this will cause an increase in blood glucose level, which will cause an increase in the secretion of insulin. And this definitely will cause rapid uptake, storage, and use of glucose by almost all tissues of the body, but especially the muscle, the adipose tissue, and the liver. There are few exceptions, and a very important exceptions: the brain, one of them, kidney, red blood cells, and intestinal mucosa, and the gonads, that what they call them insulin independent glucose uptake. So like for example, the brain does not need the insulin to take up the glucose from the blood. As we said, in the muscle, the insulin promotes the uptake and the metabolism of glucose. Usually, the muscle depend on fatty acid for energy production, not the glucose. But under two conditions, the muscle will use large amount of glucose, will uptake large amount of glucose from the blood. The first condition during exercise, the moderate and the heavy type of exercise, and the exercise itself will increase the glucose transporter's translocation, and this happen even in the absence of insulin. So that's why this mechanism explain why exercise is important for diabetic patients, because the exercising muscle will increase their uptake of glucose by increasing more of GLUT4 transporters at the cell membrane of the muscle cells, which will increase the uptake of glucose from the blood. And this is, as we said, even in the absence of insulin. The second condition, when there is after having a meal and during the postprandial phase, when there is high glucose level and the insulin is high, then this insulin will cause rapid transport of glucose into the muscle cells. And once this glucose inside the muscle, it will be stored as muscle glycogen, and it will be there in case the muscle needs it later on. What about the liver? Also, we said that the insulin will also promote glucose uptake, storage, and the use by the hepatocytes. So one of the effects that insulin stimulate glycogenesis in the liver, which means storing the glucose as a glycogen inside the liver. So once we have a high blood sugar, say for example after a heavy carbohydrate meal, then the pancreas will secrete the insulin, and the insulin will cause rapid uptake and storage of this glucose inside the liver as glycogen, which will help to reduce the blood sugar level after the meals. So how the insulin will stimulate glycogenesis? First one, it will inhibit glycogenolysis. So there will be no more breakdown of the available glycogen in the liver by inactivating the enzyme responsible for that, which is the liver phosphorylase. The second mechanism, it's by enhancing the uptake of glucose by increasing the activity of the enzyme glucokinase, which is the first step there in once the glucose go inside the cell. So this will increase more of glucose uptake by the cells. The third mechanism is by increasing the activities of the enzymes that promote glycogen synthesis, and one of them is glycogen synthase. So these are the three mechanisms where the insulin will stimulate glycogenesis in the hepatocytes. Also at the same time, the insulin inhibit new formation of glucose, which we call it gluconeogenesis. So the net effect of insulin on the liver, it will increase glycogen synthesis and it will inhibit gluconeogenesis. Between the meals where the blood glucose start to drop, because now it has been removed by either the muscle, the liver, and also the adipose tissue, as I will discuss later on. Now the liver will also help in stabilizing normal glucose level between the meals. How is that? So once the blood glucose start to decrease, the pancreas will also decrease its insulin secretion, because as we said, the most important control of insulin secretion is the blood glucose level. So once the insulin secretion is reduced, this will cause the insulin effect on the liver to be reversed. So now the enzyme phosphorylase become activated, and this will cause glycogenolysis. Also the enzyme glucose phosphatase now will be activated, and this will cause the phosphate radical to split away from glucose and the free glucose to be released into the blood. So here the liver will act as a buffer system. So it will remove the glucose from the blood when it is present in excess after a meal, and then it will return it to the blood when the blood glucose concentration falls between the meal. So it's a very important organ for glucose level control. What about the effect of insulin on fat metabolism? Insulin promotes fat synthesis and storage. How is that? What is the mechanism? First of all, the insulin will increase the utilization of glucose by the body. So most of the energy the body needs will come from glucose, so the fat will be spared and it will not be touched. The second thing, the insulin promotes fatty acid synthesis in the liver. So the liver will take the glucose, it will store it as glycogen. Once it reaches its maximum capacity to store glycogen, now it will convert the glucose into fatty acid. And this fatty acid are then transported from the liver through the blood by a way of lipoproteins, which then will go into the adipose cells to be stored there. So now the excess glucose will be converted to fatty acid, which then will be stored in the adipose tissue as fat. Also, the insulin inhibit a hormone called an enzyme called hormone sensitive lipase. This hormone sensitive lipase cause hydrolysis of the triglycerides already stored in the fat, which in another word, it causes breakdown of the fat. So the insulin will inhibit this enzyme, so there will be no more breakdown of fat. So during the fed state, when the glucose level is high and the insulin is high, the excess of glucose will be stored also as fat, and already the fat there in the body, it will not be touched. Also, insulin will increase the transport of glucose into the adipose cells. So the uptake of glucose by adipose cells also will be increased. And this will provide a substrate for glycerol. And the glycerol is a very important molecule for fat synthesis, for triglyceride synthesis, because it is important in building up the triglycerides and then the fat globule. So insulin here at the end, it will promote deposition of fat in these cells. So the net effect of insulin and fat metabolism, it increases the fat synthesis and storage. So now I want you to answer this question. Do carbs make you fat? How is the insulin might contribute to obesity? Just review the effect of insulin on carbohydrate and on fat metabolism, and you can answer this question. The brain cells is quite different from other cells of the body, in which there is a lack of effect of insulin in glucose uptake and usage by the brain. Most of the brain cells are permeable to glucose without insulin intermediation, which means it does not require insulin for the glucose to enter the cells, which is different than the other cells. As we said, they need the insulin to translocate these GLUT4 transporters so the glucose can go inside the cells. Another thing important about the brain, that the brain cells normally use only glucose for energy, and they have very difficulty in using other substrates like fats or any other things. So the main source of energy for brain cells is glucose. That's why we need to maintain blood glucose level in a normal range. Any decrease in blood glucose level, the brain cells will be affected, because now there is no energy for the brain cells because the glucose is low. And here the patient or the person will develop symptoms of hypoglycemic shock. And this is mainly the person will be nervously irritable, he might have fainting, seizures, and sometimes it might reach to coma. That's why it is very important to have a normal blood glucose level, which we should not drop beyond that critical level. Okay, what about the effect of insulin on protein metabolism? Insulin promotes protein synthesis and storage. That's why insulin is one of the anabolic hormones. What is the mechanism here? Insulin stimulates transport of amino acid into the cells. So more of amino acid will go inside the cell. This will increase, also it increases the translation of messenger RNA and more of protein formation. Also, over a longer period of time, insulin increase also the rate of transcription of selected DNA, which will increase the protein synthesis. Also, insulin inhibit the catabolism of protein. So It stimulates synthesis of protein. At the same time, it inhibits catabolism of protein. In the liver itself, insulin, as we said before, it decreases the rate of gluconeogenesis. And gluconeogenesis, which is formation of new glucose from other substrate like amino acid, which now here insulin will conserve the amino acids and it will not be used for glucose formation. So it will be spared and it will be stored in the body. Insulin and growth hormone both are very important for growth, and actually they are interact synergistically to promote growth. Also, because insulin is required for the synthesis of protein, so that's why it is essential for growth of an animal, same as growth hormone. So both of them, they are needed for the growth, because both of them, they are stimulating protein synthesis, which is important for growth. Here an experiment done on a rat. So what they did here, depancreatized, which means they removed the pancreas, and hypophysiectomized, which means remove the pituitary. So there will be no growth hormone and no insulin secreted in that rat. So now here they give only growth hormone. There is no much increase in the weight. Here they give only insulin. Again, there is no much increase in the growth. But once they give both growth hormone and insulin, the weight start to increase and the growth rate is increased. And this means that both the hormones, the growth hormone and the insulin, function synergistically to promote growth. And this happens because each of the hormones have a specific function that is separate from the other one. How the body controlled the insulin secretion. One of the important controllers of insulin secretion is the blood glucose level. So when there is a high blood glucose level, there will be high secretion of insulin. During hypoglycemic state, when there is low blood glucose level, there will be decrease in insulin secretion. The action of glucose on insulin is a biphasic, which means once the blood glucose level is high, there is a rapid phase, as we see here. So this is one, as plasma glucose concentration increased, the insulin secretion is increased. At the beginning, within 3 to 5 minutes, there is a rapid phase, a rapid increase in the insulin secretion. And then a slow prolonged phase, which is the second phase here, and this will take around 15 minutes. These are the different factors that control insulin secretion, and if you review the effects, automatically you will know how these will affect the insulin secretion. This diagram is just a summary of these factors. So any condition that increase the blood glucose level, so all the factors relating to the feeding state, food intake will increase the gastrointestinal hormones. It also usually after food, the parasympathetic stimulation is high. So all these and the increase in blood amino acid concentration after eating, all these will stimulate the beta cells to secrete insulin. During stress, because of sympathetic stimulation and epinephrine, this will inhibit beta cell and will inhibit insulin secretion. Okay, so if you go back to that table, this is just a summary of these individual factors mentioned there. What happens when we have insulin deficiency? The insulin deficiency can be either because the pancreas itself is not secreting insulin, like in type 1 diabetes, or because of insulin resistance like in type 2 diabetes. So the insulin here is deficient either in the amount or in the function. If we recall the effect of insulin, we can get how these patients of insulin deficiency will be presented. Let's see here what's the effect of insulin deficiency on carbohydrate metabolism. Now, without insulin, the glucose cannot be taken by the cells, mainly the muscle and the fat cells. So the decreased glucose uptake by these cells will cause an increase in glucose concentration in the blood, what we call it hyperglycemia. Also, because there is no insulin, the process of gluconeogenesis, formation of new glucose, and the process of glycogenolysis, which is breakdown of glycogen into glucose, will be increased, which will pour a lot of glucose in the blood, which also will cause hyperglycemia. What happens when we have hyperglycemia, high glucose concentration in the blood? The transport maximum of glucose in the renal tubules will be exceeded, so whatever more glucose filtered will be lost in the urine, in the filtrate, which will cause osmotic diuresis, because the osmosis now of the filtrate will be increased, and this will draw fluid from the body, and this will eventually will cause dehydration and electrolyte imbalance. What about of the effect of insulin on protein metabolism? Now here there is no insulin, then there will be catabolism of protein, and there will be no building up of protein. That's why a lot of amino acid will be there in the blood, which will be used by the liver to in the process of gluconeogenesis to form new glucose, and also some of them they might be as energy source for some cells, which this will increase the urea production by the body. And with the progressive time, if this condition is not treated, the muscles will be affected, and there will be wasting and atrophy of the muscles. What about the fat? When there is no insulin, the body will switch the energy from using carbohydrate to using fat to produce energy. So now here the process of lipolysis will be increased. A lot of free fatty acids will be delivered into the blood, and this will be used by the liver by the liver and the other cells for the energy, and this will result in formation of ketone bodies, and these ketone bodies will cause ketonemia and acidosis, and this, if not treated with the dehydration, the patient might reach hospital with coma. Also here, because there is no insulin, the process of lipolysis and a lot of free fatty acid delivered to the liver also will increase the production of cholesterol and triglycerides, and these will be transported in the blood as lipoproteins, and this will cause atherosclerosis and a lot of vascular diseases. The second important hormone secreted by the pancreas is glucagon. It is secreted by alpha cells of the pancreas, and this one is complete opposite of insulin. It increases the blood glucose level, that's why it's called hyperglycemic hormone, and most of its action on the hepatic cells, liver cells. Glucagon stimulates glycogenolysis and increase blood glucose concentration. Like what I told you, it is opposite to insulin. So glucagon here activate the adenyl cyclase in the hepatic cell membrane, which will cause formation of cyclic AMP, which will activate the protein kinase, which then will activate phosphorylase B kinase, and this will convert phosphorylase B to phosphorylase A, which then will cause glycogenolysis and formation of glucose 1-phosphate, which then will be dephosphorylated and released into the circulation as glucose. Another important function of glucagon to increase the blood glucose level is by stimulating the process of gluconeogenesis. So together with breaking up the glycogen to release glucose, it also forms new glucose from amino acid. Glucagon increases the hepatic extraction of amino acid from the plasma, and it increases the activity of the important key gluconeogenic enzymes. So more also of glucagon have another effect, and this is usually happen only when the concentration rises well above the maximum normally found in the blood, like activating adipose cell lipase, which will make an increased amount of fatty acids available for energy system of the body. So more of fatty acids will be released. Inhibit the storage of triglycerides in the liver, which prevent the liver from removing fatty acid from the blood. Also, it enhances the strength of the heart contractions, increase blood flow in some tissues like the kidney, and also it enhances bile secretion, and also has an inhibitory effect on gastric acid secretion. How the blood glucose regulated by the body? Again, the main important regulator is the blood glucose level. So conditions of hypoglycemia, where the blood glucose level is low, there will be more of glucagon release. And conditions of hyperglycemia will cause less glucagon release, which is opposite to insulin control. If you see here, when there is an increase in plasma glucose concentration, there is an increase in insulin secretion. While when there is an increased blood glucose level, there is a decrease in glucagon secretion. So there are other regulators might affect glucagon secretion, like blood fatty acid concentration. So decrease in fatty acid concentration will cause an increase in glucagon release. Also, an increase in the concentration of amino acid will increase glucagon secretion. And here it's the effect of amino acid similar to insulin. So increase in amino acid secretion will increase both glucagon and insulin secretion. Exercise is another factor that stimulates glucagon secretion, even when there is no fall in blood glucose level, and this can be explained because of the nervous stimulation during exercise. There will be more of a nervous stimulation to the cells to increase glucagon secretion, and also because of an increase in the circulating level of amino acid because of an exercising muscle. Here it's easily to work out the insulin glucagon ratio in different conditions. So if you recall that insulin is a glycogenic, anti-gluconeogenic, and anti-lipolytic, and anti-ketogenic. So it is mainly a hormone of energy storage. When there is an abundance of nutrients supply, the hormone of insulin will be increased to store this nutrient supply. So it's mainly abundant during feeding state when there is a lot of nutrient supply, so the energy will be stored. While glucagon, it is a glycogenolytic, gluconeogenic, lipolytic, and ketogenic hormone, which is, we can label it as a hormone of energy release. So in conditions when energy is needed, like say for example during fasting state, during heavy exercise, here the ratio between the insulin and glucagon will be low, because more of glucagon will be released at these conditions, because here the body needs energy, so it needs to break down the glycogen to release glucose, it needs to produce more of new glucose, and this is the functions of glucagon. While when we don't need an energy, like after a heavy meal, now here what will happen? We need to store more of nutrients like glycogen, protein, and fat. So here the insulin will be more released compared to glucagon. That's why the ratio will be increased. This slide shows you how important to control the blood glucose level through the interacting action of the insulin and glucagon between the liver and the pancreas. First of all, you have to recall why it is important to keep the blood glucose level in a normal range. Can you tell me why? Yes, the brain. As we said, the brain, the only fuel for brain is glucose. That's why we need the blood glucose level to be normal. So how the body will maintain this normal blood glucose level? So in conditions of high blood glucose level, say for example after a heavy carbohydrate meal, the pancreas will secrete the hormone insulin, which will increase the uptake of glucose by the liver, by the muscle, by the adipose tissue, and it will store it there as glycogen, as fat, and as protein. But when we have a prolonged fasting, when there is no energy and the body needs an energy, now here what will happen? The insulin will be reduced and the glucagon will be increased. What will do? What has been stored now, it will be break down to release glucose. So there will be glycogenolysis, gluconeogenesis, lipolysis, all this to increase the delivery of glucose into the blood to maintain the blood glucose level normal. Another hormone secreted by the pancreas is somatostatin. Somatostatin secreted by delta cells, and it is again a polypeptide hormone with a short half-life of 3 minutes in the circulating blood, again because it is not bound to plasma proteins, and its secretion stimulated mainly by increase in blood glucose and increase in fatty acid. Also increase in blood amino acid, particularly the arginine and the leucine, and in the increase in the concentration of upper GIT hormones in response to food intake. So basically, if you see all these 3 factors, somatostatin is stimulated by all factors related to ingestion of food. So after ingesting a food, what will happen? You have a high blood glucose level, high fatty acid, high amino acid, and also the upper GIT hormones will also be released. So all these will stimulate somatostatin secretion. What are the actions of somatostatins? It inhibits both insulin and glucagon hormone secretion. It decreases the motility of the stomach, duodenum, and gallbladder. It decreases both secretion and absorption in GIT. So if you see here, what somatostatin do is extending the period over which nutrients are assimilated in the blood by decreasing the secretion, the absorption, and also it decreases the utilization of nutrients by the tissue, which makes the nutrients available for longer period of time. Also, somatostatin chemically similar to the growth hormone inhibiting hormone structure, which then inhibit growth hormone release. That's all about the pancreatic hormones. Please, if you have any question, let me know by email, and hopefully I can answer your question. Thank you very much and wish you all the best.